everyone. My name is Justine Noel, and I'm a Ph.D. candidate here at the Icahn School of Medicine at Mount Sinai in New York City, investigating how prenatal exposures influence maternal and infant health. I'm excited to share my thesis story with you today, entitled Healthy Mom, Healthy Baby: The Impact of Prenatal Depression on Maternal and Infant Immunity. Pregnancy and new motherhood can be a special time in a woman's life, as displayed by cultural icon Beyoncé on stage revealing her pregnancy at the 2011 MTV Music Awards. You may feel overjoyed. There are no words that can express having a baby growing inside you. So of course you want to scream out and tell everyone. Pregnancy can mean the beginning of a new stage in a woman's life and all of the changes that this new stage brings. We talk about the obvious ones like cravings, fatigue, nausea, body shape. But what about mental health? Pregnancy can be a time of joy and stress. Research suggests that 1 in 5 women experience depression-related symptoms throughout their pregnancy. However, the actual rate might be higher since women may be reluctant to get help. Depression is a medical condition that causes feelings of sadness and loss of interest in things that you like to do. It can affect how you feel, think, and act, and interfere with your daily life and needs treatment to get better. These women are often juggling a multitude of life stressors such as pregnancy complications, as well as family and financial stress. Untreated depression can have serious consequences on not only moms, their children, and families, but society as a whole. Depression doesn't just impact moms, it can also affect your child, your offspring. Depression during pregnancy has been shown to increase premature birth, or birth before 37 weeks of term, learning, behavior, and developmental problems, as well has been attributed to contributing to low birth weight. But did you know that depression can promote allergies in your offspring? Prenatal depression has been associated with enhancing your child's risk of developing allergic-mediated diseases like eczema, as well as food allergies and wheeze. So how does prenatal depression evoke such a disease? One hypothesis is that the maternal immune system during pregnancy may play a role. Before we jump in, let's take a step back and have a brief overview of what is known and what is not known about the mom's immune system during pregnancy. We will first look over how cytokines, special immune modulating factors, contribute to changing the immune environment during pregnancy, and then look at the impact this may have on influencing allergy development. During pregnancy, major adaptations occur in a mom's immune system to protect the mother and her future baby. It's thought that a balance between inflammatory versus tolerant immune signatures defined by cytokines play a role in influencing this state. The source of these cytokines that regulate tolerant signatures, displayed in green, corresponds to a special subset of T cells named Th2 memory and T regulatory memory cells. And these cells are seen at high frequencies during the 2nd and 3rd trimester and have been associated with the maintenance of maternal fetal communication, normal fetal development, and full-term healthy pregnancies. It's unclear the role depression plays in influencing such of a balance. However, we speculate that depression may promote inflammation, and this is due to the enhanced frequency of preterm birth observed within depression moms. And this inflammation is known to impact the fetal communication, maternal fetal communication, leading to potentially adverse fetal development and adverse pregnancies. But how does disrupting this depression pathway lead to allergies in babies? Mother and fetus are interconnected. If an exposure to these signals can lead to an adverse infant outcome like allergy development, how does depression influence this? We speculate that depression targets the maternal immune balance to skew the scale of maternal immunity, and that exposures to these cytokines during fetal immune development can influence the development postnatally and impact their allergy health outcomes, promoting either allergy response, indicated in orange, or allergy tolerance, indicated in green and seen in children who don't develop allergy-related diseases like eczema. By understanding how exposures impact maternal fetal immunity, we aim to better understand how maternal immune influences impact infant immunity and health, as well as potentially identify immune pathways involved in early allergy manifestation, all in the hopes to create new therapeutics for early interventions to prevent allergic disease. So how do we go about answering such a question? Using human blood samples from moms and their newborns based here in the city that never sleeps, New York City, we aim to answer, does prenatal depression modulate maternal immunity to influence allergy outcomes? And to address this overarching question, first we looked at two simple questions. Does prenatal depression alter maternal immune balance? And does prenatal depression impact infant immunity? Again, we took blood from our human cohort. And in the first part of this talk, I'm going to focus on the impact that depression has on mom's immunity. So we took blood from around 82 women here based in New York City, isolated immune cells from their blood, and stimulated them with various different stimuli indicated in green, blue, purple, and pink. Each of these stimuli represents or mimics the response to encounters like bacteria, which indicates the LPS response, or mimics a response to viral infections, indicated in purple. Single-strand RNA targets innate receptor TLR7, as well as T-cell responses using PMA iomycin to get a broad overview of how different exposures impact maternal immunity during pregnancy. And using a machine learning algorithm called elastic net regularization, we predicted which cytokine signatures associate with our outcome, which in this case is depression. So what is elastic net regularization, and how do we use this machine learning algorithm to predict which cytokines associate with depression? Well, elastic net regularization incorporates two penalization approaches to create predictive models that are easy to interpret. In simple terms, we'll take our cytokine data generated from our stimulation experiment assays. It assigns them weights based off their probability of predicting depression, and then it ranks these values based off of their weights and how they contribute to the overall model. To verify how accurate our model is, we used a cross-validation resampling approach. There are two ways to look at this. The first that I'm going to show is an ROC curve, or a receiver operating characteristic curve, a graph showing the performance of our classification model. Using specificity and sensitivity terms, it provides an aggregative measure of performance across all samples. This model is able to predict accurately for depression or no depression 91.7% of the time. In the graph on the right is a confusion matrix. Let's walk through this. Here I'm showing a simplified version of how a confusion matrix is created. We have our predictive elastic net model on the top and our actual results on the bottom. Highlighted in red here are our true negative, TN, or true positive, TP characterizations. And as you can see, in both elastic net and our actual results, these two outcomes align with each other. Meaning, in the true positive case, what you predicted as positive actually was positive in the results, or in a true negative case, what you predicted as negative actually came out as negative in our prediction models. As opposed to false negative and false positive rates highlighted here in purple. False negative represents a type 2 error, meaning that you predicted a negative result when actually this was false, versus a false positive error, which represents a type 1 error, is where you predicted something being true is actually false. You want these values to be as low as possible in your model, as this will allow your model to be highly predictive and accurate, as well as well balanced to predict both outcomes. Now back to our matrix. Our model was able to correctly predict depression with 116 samples being classified correctly and less than 15% or 14 samples being classified incorrectly as false positives. And as you can see on the true negative side, we see a similar ratio, highlighting how balanced our model is at both predicting depression and non-depression in our maternal cohort. So what were the differences in these samples? Here I'm showing our variable importance plots. This is showing the top cytokines highlighted in orange all corresponded to our T cell assays, correctly predicting depression in these moms, suggesting that depression may be influencing the immune balance during pregnancy. Looking at the defined variables that were isolated from our elastic net regression model, we saw that women with depression, highlighted here in the green boxes, displayed enhanced production of inflammatory associated cytokines, interferon gamma, IL-17F, and IL-17A, even after adjusting for factors such as depression medication use, race, and maternal atopy status. The level of these cytokines display a positive relationship as well with clinical depression, anxiety, and perceived stress scores obtained from the same woman, indicating that there is a relationship between inflammation and mood disorders during pregnancy. To further verify depression is associated with changes in T cell responses, we took a subset of maternal samples and profiled them using flow cytometry to get the frequency of the different T cell subsets indicated in this balanced chart here. We used a transcription factor called FOXP3, as this has been shown to be a clear identifier of a special subset of T cells called Tregs. T regulatory cells are major factors that maintain immune tolerance by suppressing inflammation. We observed that in our depression cohort in green, there were less positive FOXP3 cells, meaning that there were less Tregs in circulation, and more FOXP3 negative cells, or more effector type cells, in circulation, overall suggesting that T cell immunity is influenced by depression in these moms. We unfortunately didn't find any immune predictors of eczema within maternal immunity, so we moved into profiling newborn immunity. Taking a similar approach, we used core blood samples from matched mothers within our cohorts, and this core blood represents at-birth immunity. From this core blood, we isolated immune cells and subjected them to our stimulation assays, where we probed with different innate and T cell stimuli, as well as continued to use an elastic net regression method to identify the factors that were predictive of depression during pregnancy in infants, as well as childhood eczema, to identify potentially pre-symptomatic indicators. Within our infant immune stimulation assay, we again used the elastic net approach to identify factors related to depression exposure or eczema development in infants. Here are their respective confusion matrix to show how accurate our predictive model was. For infant immunity at birth, our classification model had some biases. It favors accurate classification of children who were not exposed to depression or children who did not develop eczema, as indicated by their low false negative rates. The Venn diagram highlights the cytokines that displayed at least a 20% influence onto our classification models. For depression, 3 cytokines related to innate immunity and antiviral response were identified. And our univariate analysis highlighted that children not exposed to depression had a higher antiviral response, suggesting that maternal depression may alter antiviral immunity postnatally. For eczema, 2 cytokines showed strong importance in our classification model, one being IL-5. IL-5 is a cytokine involved in the allergy response, suggesting that levels at birth may be a pre-symptomatic indicator of T cell dysregulation leading to allergy development. The shared factor observed between both depression and eczema outcomes in infant immunity was IL-6, which is a pluripotent cytokine with numerous roles in regulating immunity, and we're currently exploring its impact on infant immunity. Overall, our analysis here highlights that there are pre-symptomatic factors that can be observed at birth and point at different pathways and cell types that may be involved in promoting allergy disease manifestation. So let's put the pieces together. So in summary, I've shown that using elastic net machine learning technologies, we can better characterize the role of mood exposures during pregnancy, that depression relates to a reduced tolerance associated cytokines and cellular frequencies during 3rd trimester, shown by the increased production of inflammatory cytokines like IL-17A, 17F, and interferon gamma, and a reduced frequency of circulating T regulatory cells in the periphery of these moms during the 3rd trimester. I've also showed that there are signatures that relate to allergy at birth, and that can be used to predict infant eczema outcomes and points to potentially early dysregulation in T-cell immunity. Our next steps is to use a network analysis and causal interference models to estimate how does this inflammation influence either infant immunity and how does this relate to eczema outcomes. Causal interference will allow us to better understand the how behind the why. It allows us to incorporate and look at influences of additional factors like race, household income, support at home, and discrimination, and how this all plays a role in modulating this depression-related immunity that we've observed within our cohort. Overall, more work needs to be done in this area to find better intervention methods to better support moms-to-be, as well as analysis and additional cytokine signatures observed can be predictive factors in identifying women who develop potentially postpartum depression. Research in this field is critical to further understand the immunology of pregnancy and its influence on promoting prevention disease. Improving the well-being of moms, infants, and children is an important health goal. Their well-being determines the health of the next generation and can predict future public health challenges for families, our communities, and the healthcare system in general. Don't forget that support is a community effort, and it's important to remember if you or your loved ones are experiencing symptoms of depression during pregnancy to seek help to learn and share positive coping strategies to manage your symptoms. And with that, this brings me to the end of my talk. And science is a group effort, and I'm so thankful for everyone that has been involved in this project and helping it to move it forward, including my lab, my mentor, Cecilia for all advice, Danny, postdoc in my lab, who has been so instrumental for helping me to get some of these assays up and running, collaborators for their assistance with sample collection, additional collaborators in Samson's lab and Mosman's labs for analysis assistance, my thesis committee for assistance and direction, Mount Sinai for being an amazing environment to work in, as well as the iBiology staff and supporters who have been very instrumental in helping us create this series. And of course, I want to thank you all for engaging with this video, and I hope it was helpful and useful for you.